Welcome to today's Explainer. We're jumping straight into an absolutely fascinating journey to the very core of what makes you, well, you. We are going to trace the path of life, literally starting from the microscopic instructions locked tightly inside our DNA, all the way up to the formation of a developing human being. So, let's get right into the blueprint of life. Here's our roadmap for this Explainer. We'll kick things off with the blueprint of life and the rules of inheritance. From there, we'll dive into dominance and blood groups, look at determining genetic sex, and wrap up with fertilization and early development. Let's do this. Section 1. The Blueprint of Life, Understanding the Microscopic Foundation. Okay, to really grasp our genetic makeup, we first have to look at chromosomes. These are essentially long, tangled, thread-like structures made of DNA, sitting right there in the nucleus of our cells. I always like to think of chromosomes as the really thick chapters in an instruction manual. And genes? Well, genes are the specific, absolutely critical sentences within those chapters. Basically, a gene is a distinct segment of DNA that codes for a very particular characteristic. It's the exact instruction that determines something like, say, your height. Now, what's really fascinating here is the sheer contrast between the hidden code you carry and what the world actually sees. Your genotype is your internal genetic composition. It's the raw data, often written out as letters, like a capital B, or maybe a capital B, lowercase b. But your phenotype, that's your external physical appearance. It's the actual visible expression of that hidden genetic code. And this perfectly illustrates a fundamental difference in how our cells are packaged. Diploid cells, which we represent as 2N, are your standard body cells, or somatic cells. They hold a full set of paired chromosomes, one from your mom and one from your dad. But haploid cells, represented as just an N, are the gametes. These are the sex cells formed by meiosis in the testes and ovaries. They carry exactly half the chromosome number, meaning they are perfectly prepped to combine and create a brand new full diploid set. Moving right along to Section 2, the rules of inheritance. How the instruction manual is actually read. So we know we have these genetic sentences, right? But how do they interact when they finally combine? Well, if a gene is the fundamental sentence that codes for a trait, alleles are the different translations or variations of that exact same sentence. They're basically two or more versions of a gene located at the exact same position, which we call a locus, on a chromosome pair. For example, if you have a gene determining a plant's height, you might have one allele coding for tall and a totally different allele coding for short. The really crucial part is how these alleles pair up in your genotype. If you happen to inherit identical alleles from both of your parents for a single trait, say two dominant alleles for tallness, capital T, you are what we call homozygous. But if you inherit a mix, like two different alleles for that single trait on the same locus, maybe a capital T and a lowercase t, then you are heterozygous. And that brings us straight to the foundational rules defined by Gregor Mendel, which mathematically explain exactly how we inherit these traits. First up, Mendel's principle of segregation. This tells us that an organism possesses two factors, or alleles, which separate independently during gamete formation, so each sex cell only gets one. It's a clean split. Second is the law of dominance. This states that when two different alleles are present in a heterozygous individual, the dominant allele is going to completely mask or suppress the recessive one. The dominant trait basically hogs the spotlight and becomes the visible phenotype. All right, on to Section 3, Dominance and Blood Groups, the rules of multiple alleles.
So Mendel's basic law of dominance is a fantastic starting point, but let's be real, biology isn't always perfectly black and white. We actually see three distinct types of dominance. Complete dominance is what we just talked about. One allele fully dominates and the recessive one is entirely masked. But then we have incomplete dominance. This is where neither allele is fully dominant over the other, so you end up with a blended intermediate phenotype. And finally, codominance. This is when both alleles fight for the spotlight and are equally fully expressed in the phenotype at the exact same time. No blending, just both showing up.
Human blood grouping is such a great example of this because it's the result of multiple alleles. Instead of just two options, we actually have three different alleles at play here: IA, IB, and a lowercase I. Now, blood groups A and B show complete dominance over that recessive lowercase I. But if you inherit both IA and IB, they don't blend, and neither masks the other. Instead, we get codominance, which results in the AB blood type. On the flip side, if you inherit two recessive I alleles, you get type O blood. To really lock this complex system into your memory, use this incredible, simple guide. the 1, 2, 3, 4 rule of blood. It goes exactly like this. An individual has exactly one blood group phenotype. They possess exactly 2 alleles for that blood group in their genotype. Those 2 are selected from 3 different possible alleles circulating in the broader population, and all those combinations ultimately result in exactly 4 possible human blood groups: A, B, AB, and O. Pretty neat, right? Section 4. Determining genetic sex. Reading the final chapter of chromosomes. When you look at a full karyotype, it lays out all the chromosomes found inside a somatic cell. And you'll notice how the first 22 pairs, our autosomes, are virtually identical in males and females. But that final pair, the gonosomes, that changes everything. Females possess 2 X chromosomes, while males possess 1 X and 1 Y. That single specific genetic combination is what determines biological sex. Finally, Section 5, fertilization and early development, going from the blueprint to the building. We've spent this time examining the microscopic code, so now let's actually watch the blueprint spring into action. The development of a human being kicks off with fertilization, where a haploid sperm unites with a haploid ovum. This monumental event creates a single diploid cell called a zygote. After that, this newly formed zygote goes on a rapid journey of division. It transforms into a multicellular embryo, progressing through stages like the morula and the blastocyst before eventually maturing into a complex fetus. I want you to imagine time just speeding up right before your eyes. Almost immediately following fertilization, that single-celled zygote, which is carrying that newly combined diploid genome with all those dominant and recessive alleles we just covered, begins the process of mitosis. It splits perfectly in half to form the 2-cell stage. And that rapid pace, it doesn't stop. The cells just keep doubling, dividing again to create a highly distinct 4-cell stage. And all the while, that precious genetic blueprint is being faithfully replicated and tucked securely inside the nucleus of every single newly formed cell. After continuous, non-stop rounds of division, the structure eventually forms a tightly packed cluster of cells known as a morula. From there, it develops into a hollow ball of cells called a blastocyst. This intricate, microscopic structure is now fully prepped for implantation, where it'll embed itself into the nutrient-rich lining of the uterus, and the incredible, long process of gestation truly gets underway. Now, before we wrap up, we do have to acknowledge something. This incredibly intricate process of copying DNA during cellular division isn't always absolute perfection. Sometimes a sudden change occurs in the sequence or order of the nitrogenous bases within the nucleic acid. In the world of genetics, we call this a mutation. And I want to leave you with a bit of a provocative thought today. We know that genetic variation relies heavily on the mixing of maternal and paternal genes to create entirely new genotypes and phenotypes. But considering the sheer complexity of our blueprints, could it be that mutations, these sudden, spontaneous changes in the genetic code, are not just biological errors? What if they are the very engine of evolution itself, providing the novel traits necessary for a species to adapt, survive, and thrive? Thank you so much for joining me for today's explainer. Keep questioning the incredible biology all around you.